Okay, so welcome back to this next video in which we are discussing personalized cancer medicine. Okay, so we're in the process of discussing some examples which have a taste of the principle of personalized cancer medicine within them. Okay, so we've seen so far the thiopurines, tiaguanine and 6-mercaptopurine, and before they are administered, what happens is that genetic tests are done to examine the genes for the TPMT enzyme, okay? And this is because the TPMT enzyme is involved in deactivating those drugs, and certain polymorphisms that are found in the normal healthy human population of TPMT cause deficiency in the activity of TPMT, which isn't disease-causing until you give someone this drug or one of these drugs. Okay, so basically what we do is we tailor the dose to your TPMT activity, basically. Okay, and then there's a similar story for capecitabine, which is a prodrug for 5-fluorouracil, okay, and this is broken down by the enzyme dihydropyrimidine dehydrogenase, which is found within hepatocytes. And again, certain polymorphisms that are found within the normal healthy human population result in lower activity in DPD, known as DPD deficiency, and then that's going to mean that when you take the 5-fluorouracil, if you have DPD deficiency, then the drug is going to remain within your body for a much longer time, okay, and that's how you can get very adverse reactions to the drug. And then obviously the solution to that is reduce the dose for these people with DPD deficiency. So again, it's an example of personalizing the treatment to your genetics, okay, it's not quite yet to your entire genome, okay, that's still far off. In fact, all of the examples that we're going to see, they're really concerning one gene. Okay, right, so irinotecan is the next example that I want to talk about. Okay, so irinotecan is again an example of classical chemotherapy. All of the drugs at the moment that we're looking at, capecitabine and the thiopurines and irinotecan, they're all examples of classical chemotherapy. We'll come on to some more targeted chemotherapy agents later on. Okay, so irinotecan then. Irinotecan is used to treat lung cancer and also colorectal cancer, so lung and colorectal cancer, okay, and the way it works is quite interesting. It inhibits what is known as type 1 topoisomerase enzymes. So I want to explain to you what it is that type 1 topoisomerase enzymes do. So basically this is a really important concept, okay? So whenever you want to make more DNA or make more RNA, you have to unravel the DNA, okay? So let me draw a picture of this. So I usually draw DNA as just two parallel lines, but in reality, of course, what we know is that in fact the DNA is coiled up into this double helix like so. Okay, now what's going to happen is when you want to make RNA from a gene, or when you want to copy the DNA, okay, so remember those are the two key processes in cell division. In cell division you need to copy the DNA and you need to make loads of new proteins, so you need to make lots of RNA. Okay, so these two key processes, replication of the DNA and transcription of the DNA, these require you to open the DNA up. So what's going to basically happen is you're just going to push the coiling along. Okay, so what is going to happen is you're going to uncoil these portions here, and what then is going to happen is up here you're now going to get it really coiled up very densely like this, so twisted up very densely. Okay, so you end up increasing the twist here. Okay, and that's going to happen whenever you either transcribe this piece of DNA here or replicate the DNA here, so increasing the twist. Now, what this can result in is it can result in the DNA breaking. It can also result in what's known as supercoiling first. So there's a piece of terminology that people use with regards to this, which is supercoiling. Okay, so I want to try and explain to you what supercoiling actually means. And to do this, I have a prop, okay, and I want to make sure this is actually seen on the camera. So what I've made is just two pieces of paper, okay? Those are long and I have cut up. Okay, and these are going to represent our two strands of DNA. Okay, so here they are, and at the moment they're not twisted together. So what I'm now going to do is I'm going to twist them up a little bit. So I'm going to hold these two together here, and then I'm going to start twisting one around the other. Okay, like so. I'm hoping to show you the phenomenon of supercoiling. Okay, so here we go. I'm twisting the pieces of paper around one another, and you can of course do this at home. Okay, and now, this is probably good enough now. Here, I'm holding the DNA taut at the moment, like so, but what happens if I sort of let it go a little bit? Okay, maybe I haven't put enough twist in. Let me twist it a bit more, and then then it will hopefully supercoil. Okay, so twist, 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 twist. Okay, right. Now it's going to do it. Okay, so when I'm holding it taut, I can hold it like this, but then if I let it go, look, the whole thing starts to coil up in a bigger way now. The whole DNA double helix starts to coil in this bigger way that you can see here. Look, the whole thing is coiling. That is the concept of supercoiling, basically. And when you increase the twist of the DNA here, there's the risk that it will supercoil just like this. And there's the risk that it will then start to break. Okay, in addition, if you make the twist too dense here, eventually you just can't push the DNA further anymore. You can't push on with your transcription or your DNA replication because the twist is just too dense here. Okay, and you just can't make it any more dense. Okay, so eventually what can end up happening is DNA replication will stall, transcription will stall. Okay. So, what is the solution that the cell has to this? Well, it has enzymes known as topoisomerase enzymes. Okay, now there are type 1 topoisomerases and there are type 2 topoisomerases. Type 2 topoisomerases are rather complicated to explain what they actually do. Okay, and thank God we don't have to go there because irinotecan doesn't inhibit the type 2 topoisomerases. It works on the type 1 topoisomerases. Okay, type 1 topoisomerases, it's far easier to explain what they do. Literally, what they do is they go to the DNA, they snip one of the strands, okay, and they then undo one of the twists. So they will pass the strand back around the other strand and they will remove one of the twists, or multiple twists. They can do more than one. Certain type 1 topoisomerases can undo more than one at once, okay, but many of them will just undo one twist, okay, and that's therefore how they reduce the twist here. Okay, and therefore they allow transcription and DNA replication to occur. So without these essential enzymes which remove this very dense twisted portion upstream of replication forks or transcription forks, okay, you cannot transcribe your DNA and you cannot replicate your DNA. So this is very, very important, basically. And irinotecan works by blocking these type 1 topoisomerase enzymes. And by blocking those, you stop this from occurring, or at least you reduce the cell's ability to cope with this, okay? And therefore what you do is you reduce the ability of a cell to replicate its DNA, and you reduce the ability of a cell to transcribe its DNA and produce RNA. Okay, and therefore that is the basis of how irinotecan then leads to problems with cell division and how it can stop cells from dividing. Okay, right. Now, one of the things that I should say is that irinotecan, the actual molecule, is actually a prodrug. It's not the molecule that actually does block the type 1 topoisomerases. What it is first processed to is something known as SN38, and this is the active drug that will actually inhibit the type 1 topoisomerases. Okay, then what happens is SN38 is then deactivated again by a liver enzyme. Okay, so SN38 will then go to the liver and be deactivated, and it will be deactivated by an enzyme with a fantastic name known as UDP and then glucuronide transferase 1A1. So glucuronide and then transferase, is it going to fit in? Okay, no, just glucuronide transferase and then 1A1. And now for short, this thing is abbreviated, thank God, down to U for UDP, G for glucuronide, and then T for transferase, 1A1. Okay, so 1A1 just refers to the specific UDP glucuronide transferase here. Okay, so there are many different glucuronide transferase enzymes. This is specifically 1A1. Okay, so now it's the same phenomenon as we've seen for capecitabine and for the thiopurines. This UDP glucuronide transferase 1A1 has polymorphisms that are seen within the healthy human population. Okay, and specifically one of the polymorphisms that can occur in this enzyme is known as UGT1A1, and then for some reason you put a star and then 28 here. So this refers to a specific allele of UGT1A1. Okay, now if you have this allele of UGT1A1, what it results in is much lower production of the UDP glucuronide transferase 1A1, and that then results in the liver's UGT1A1 activity being reduced. So the liver's ability to process the SN38 and deactivate it is therefore reduced. So therefore, when you take this drug, the drug will remain active in the blood for much longer, basically. Okay, and that can result in very serious side effects. This can also result in bone marrow suppression if it's given at either a too high a dose, or if you have this UGT1A1 deficiency that then blocks it from being broken down properly. Okay, so this can also cause bone marrow suppression by blocking the cell division within the bone marrow that is needed in order to make new red blood cells, white blood cells, and thrombocytes. Okay, right, so again what happens is before prescribing this drug, tests for healthy function of UGT1A1 is performed. Okay, so again this is an example of where we can look at your genes and predict whether you are going to have a horrendous response if given a normal dose of irinotecan. Okay, right, so those are the ones now where purely what this has been about is pharmacokinetics. Okay, this has all been about looking at genes to understand whether someone is going to have a horrendous response to a drug because of problems with breaking the drug down, and therefore the drug remaining in the circulation at too high levels for far too long. Okay, what we're now going to move on to is looking more at drugs which we use only when the tumour actually has a certain phenotype. Okay, so we only use them when the tumour actually has a phenotype that allows them to work. Okay, now the first example that I'm going to give you of this actually is interesting because it not only has a pharmacokinetic aspect to its pharmacogenomics, but it also has the pharmacodynamic aspect to its pharmacogenomics. Okay, and this is the example of tamoxifen. Okay, which is used to treat certain types of breast cancer. Okay, but you don't just give it for all types of breast cancer. So this is a new idea that we've got here. This is going over to the pharmacodynamics aspect here. All the other drugs that we've looked at so far, thiopurines, capecitabine, and irinotecan, okay, those were just classical chemotherapy agents which just stopped division in whatever cell they went into, basically. Okay, and the pharmacogenomics to that was trying to work out who's going to have the horrendous responses because of problems with pharmacokinetics. Now, tamoxifen has a pharmacodynamic aspect to its pharmacogenomics in that it's not actually effective in all forms of breast cancer. The breast cancer has to have certain molecular phenotypes, okay, which we'll discuss in a moment. So let's firstly, before we see which forms of breast cancer tamoxifen is actually used in, let's see how tamoxifen works. So tamoxifen again is a prodrug. Okay, and it has to be activated by an enzyme that is within the liver to something known as endoxifen. Okay, so tamoxifen is activated to endoxifen by an enzyme in the liver which is a cytochrome P450 enzyme and therefore has the prefix CYP. So CY is for cytochrome, okay, and then the P is for P450. So this is a cytochrome P450 enzyme, and specifically the one that does this is cytochrome P450 2D6, which is just its code name, basically. Okay, so this is a specific cytochrome P450 enzyme that is present in the liver. Okay, so this liver enzyme is going to activate the tamoxifen to endoxifen. And now what does endoxifen do? Well, it's an antagonist for a certain receptor. Okay, and specifically the receptor that it's an antagonist for is the estrogen receptor. Okay, and I apologise to my American viewers for using the ridiculous British English spelling of estrogen. If you are American, just wipe that silly O off. Okay, I can't help it. Estrogen receptor. Okay, right. Now, what is really interesting about endoxifen is that it's actually only an antagonist for estrogen receptors in breast tissue, not estrogen receptors expressed in other portions of the body, okay, notably the ovaries, where in fact it's actually an estrogen receptor agonist, but that's a different story. Okay, so estrogen receptors in breast tissue, not in other portions, okay? Right, so how does this then help in breast cancer? Well, the answer is it only helps in certain forms of breast cancer. Okay, certain forms of breast cancer rely on the estrogen receptor for division. Okay, so certain forms of breast cancer, so I'll just draw a picture of a tumour so that I have something to point at. Certain forms of breast cancer, and let's say this form of breast cancer is one of the ones that's going to be sensitive to tamoxifen. Okay, certain forms of breast cancer rely on the estrogen receptor for division. Okay, so in the cytoplasm of cells, there is a receptor, which I'll just draw as a box, which is the estrogen receptor. And I'm not going to be able to fit in its name into the box, never mind, I'll put it below here. So this is representing the estrogen receptor. Now, when estrogen binds to the estrogen receptor, so here comes estrogen, what will happen is the estrogen receptor will then translocate into the nucleus, and it acts as a transcription factor and changes gene expression. And in certain forms of breast cancer, the activity of the estrogen receptor is absolutely essential for these cells to be dividing. Okay, and if you stop the activity of the estrogen receptor, the cells will stop dividing. Okay, but that's only in certain forms of breast cancer. And these forms of breast cancer where the cancer cells are utterly dependent on estrogen activating estrogen receptors to divide are known as estrogen-dependent forms of breast cancer. Okay, so estrogen-dependent breast cancer. Okay, and in these estrogen-dependent forms of breast cancer, if you then give a drug which blocks the estrogen receptor in breast tissue, such as tamoxifen, which is then activated to the active compound endoxifen, then you block the estrogen receptor's activity. So what will happen is endoxifen, which I'll draw here, will basically come and bind to the estrogen receptor. So this is our drug here in red. It will come and bind to the estrogen receptor in breast tissue, okay, in these breast cancer cells. And it will then block the estrogen from being able to bind to the estrogen receptor. That's the meaning of an antagonist, okay? But it will not stimulate the estrogen receptor. It will bind where the estrogen would bind, but it will not actually stimulate the estrogen receptor to go into the nucleus and change gene expression in the way that estrogen would. Okay, so it just blocks estrogen from being able to activate the estrogen receptor. Okay, and that effectively turns off then the activation of the estrogen receptor. And in these breast cancer cells, which are utterly dependent on the activation of the estrogen receptor in order to be dividing, that then turns off their division signal and they stop over dividing. Okay, and that's a very useful drug in certain forms of breast cancer. Only the forms of breast cancer where the cells are actually estrogen dependent. And to work out which forms of breast cancer are actually estrogen dependent, what you have to do is take a biopsy of the breast cancer cells, okay, and you have to look at the tumour genome and work out whether it is estrogen dependent or not, okay. So this is an example of where you're looking for a pharmacodynamic property, basically. You're looking to see whether the drug target is actually important to the cancerous phenotype in the breast cancer cells to determine whether the drug will have any efficacy against the tumour. Okay, this is a very different approach from the one-size-fits-all. This is looking at each individual patient and working out whether the drug will actually work and achieve an effect, rather than just giving it and hoping by trial and error that it might actually work. Okay. So, the other interesting thing about this one's got a very rich pharmacogenomic profile, okay, is that some people also have problems with cytochrome P450-2D6. Okay, so again, this enzyme in the liver, this enzyme has multiple polymorphisms in the human population, okay, and certain polymorphisms in the cytochrome P450-2D6 gene result in lower activity of this enzyme in the liver. Okay, what's known as cytochrome P450-2D6 deficiency. Okay, now this is a very different case to all of the other deficiencies that we've seen. If this time you have deficiency in this enzyme, it's not going to lead to horrendous side effects of the drug. In fact, it's going to lead to the drug being ineffective, okay? Because, remember, tamoxifen had to be converted to endoxifen to actually have an effect. Okay, so if you've got deficiency in this enzyme, it's going to mean that the drug isn't actually going to have that much effect because it's not going to be converted to the active compound. Okay, in addition, it can actually lead to side effects of this drug as well. Okay, so I was telling a little bit of a lie. Because if tamoxifen isn't converted to endoxifen, the tamoxifen can go and often have other effects, basically, and this can lead to side effects. So, this one's very, very interesting. So, cytochrome P450-2D6 deficiency, it's going to firstly result in the drug being ineffective, well, less effective, because you're not converting as much of the tamoxifen to endoxifen, which is the active compound. In addition, it can lead to increased side effects because the tamoxifen can go off and have other effects now that it's not being broken down and converted into endoxifen. Okay, so there's a very rich personalised medicine component to the prescription of tamoxifen for breast cancer, okay, and it's got both these pharmacokinetic aspects and the pharmacodynamic aspects. Okay, right, so the next drugs that we're going to turn our attention to are now going to purely have the pharmacodynamic aspect here rather than the pharmacokinetic aspect. Okay, so a drug that you cannot do a video on personalised cancer medicine without mentioning is Trastuzumab, a very famous drug. Okay, so Trastuzumab. Now, it's infamously known by its brand name, so I'll give you that as well. Its brand name is Herceptin. Now, this is used in certain forms of breast cancer, and again, just like tamoxifen, there are only certain forms of breast cancer that this is going to be effective in. Okay, so let me explain what it does, and then we'll see how this is only going to be effective in certain forms of breast cancer. And really what's key here is the concept of inter-tumour heterogeneity, the fact that different breast cancers are not the same, basically. They have different molecular properties that are actually leading to the cancerous phenotype. Okay, right. So, what is Trastuzumab firstly? So the first thing that I should mention is that it's what is known as a monoclonal antibody. Okay? Any drug ending with the suffix mab. Okay, let me just highlight this up. So the mab suffix, that stands for monoclonal, that's the M, antibody, that's the AB. Okay, so any drug ending with mab, instantly you know it's a monoclonal antibody. Now what does this mean? It means that basically it is an antibody targeted against a specific epitope of a certain molecule. So let me just remind you of a little bit of basic immunology here. So this is the basic structure of what an antibody looks like. You have two heavy chains, okay, which I'll highlight in blue. So these are the heavy chains of the antibody, and those are linked by disulfide bonds here. Okay, so these in blue, these are two heavy chains. Okay, then you have two light chains which are attached to the two heavy chains. Okay, so in turquoise here, that's one light chain, and here then is another light chain. Now, most of the antibody structure is conserved between different antibodies. Okay, however, this portion up here and this portion up here, these are different for different antibodies, basically. Okay, and they're going to be identical. These two sites on the antibody are going to be identical, and what they do is they bind to some other protein. Okay, and the meaning of monoclonal antibody means that not only do these antibodies bind to a certain protein, but they bind to a specific epitope, okay? And to really understand that, we need to be thinking not just about one antibody, but a mixture of antibodies. So let me just draw a few beakers out here. Okay, so here's one beaker, okay, and here is another beaker. Okay, so basically, let's say we want to make antibodies against a certain protein. So let's say we've got protein A here. So let's just say this is protein A, and I don't mean to imply that it's protein kinase A or anything. It's just some arbitrary protein. Okay, and we want to make antibodies against it. Now, how is the classical way that we make antibodies? Well, what we do is we go to some animal and we inject this protein in, and their immune system will make the antibodies for us. However, when you do that, what you'll end up with is antibodies which bind to all sorts of different portions of this protein A. Okay, so let me show this. So if we have this mixture of antibodies here, okay, like so, these antibodies are not going to be identical, okay? Well, some of them in the mixture will be. They'll just happen to be the same, but others will be very different. So let's say this is one antibody that binds to a certain region of this protein. Okay, I need to color code this to make it make sense. So this portion in orange here, okay? This will bind to a certain little portion, let's say this corner here of this protein A. Okay, so here's the heavy chain, like so. Okay, and this special portion of protein A that it binds to is known as the epitope, which this antibody is specific to. Okay, so this antibody will bind to a certain epitope of protein A. Okay, so that's what orange antibody binds to. Okay, and then the other antibody that's also in this mixture, which we'll call the green one here, will also bind to protein A. So they're both against the same protein, however it will bind to a different epitope, so it will recognize a different little portion of this protein here. Okay, so here is antibody green bound here. Okay, like so. So this concept here is what's called a polyclonal antibody against protein A. So this mixture is referred to as the polyclonal antibody. So this is the first confusing thing, that people use the term antibody to mean a solution containing loads of antibody molecules against protein A. But they also use antibody to mean the individual antibody molecule. That's the main confusion here, okay? That polyclonal antibody refers to the solution containing loads of antibodies that all bind to protein A, but which bind to different epitopes. Monoclonal antibody refers to a solution containing only one type of antibody, i.e. antibodies that all bind to the exact same epitope of protein A. Okay, so here, this solution here, where they're all antibody oranges, let's say, and they bind to a specific epitope of protein A, that's what's meant by a monoclonal antibody. So Trastuzumab is a mixture of loads of antibodies that are all identical and bind to a certain epitope of a protein, basically. So this specific domain that all of these antibodies have binds to a specific epitope of some protein. Okay, so that's the difference between a monoclonal antibody and a polyclonal antibody against a protein. So, Trastuzumab then is a monoclonal antibody against a protein known as HER2. Okay, now what does this stand for? HER2 stands for the human, that's the H, and then epidermal growth factor, EGF. Okay, so I probably should just write that out in full at least once. So this stands for epidermal, that's the E, and then whenever you see GF in biology, usually it stands for growth factor, and I'm sorry for writing growth. Okay, imagine that that's an O rather than an A. Growth factor, in fact I will cross it out and replace it with an O. There we go. Epidermal growth factor, so human epidermal growth factor, okay, and then receptor, and it's specifically the human epidermal growth factor receptor 2, HER2. Okay, now there are other HERs. There is actually HER1, HER3, and HER4 as well, so there are 4 HERs. Okay, we've specifically made a monoclonal antibody that binds to a specific epitope of HER2. Okay, so let me show this. So HER2 is a receptor on the surface of cells. So let's say this is the cell membrane here. These 2 parallel lines now no longer represent a double-stranded piece of DNA. Instead, they represent the inner, the bottom line, and outer leaflets of the phospholipid bilayer. Okay, and sitting in the phospholipid bilayer, you'll then have your HER2 receptor here, which we'll colour in a special colour. So HER2 can be coloured in blue here. And then Trastuzumab, or Herceptin, is a monoclonal antibody that binds to some epitope on the extracellular domain of the HER2 receptor. Now it's very important that you understand that it has to bind to an extracellular domain because antibodies are huge, great molecules. They have not a chance in the world of getting inside the cell. Okay, so they can only bind to extracellular domains of the protein. So here is our Trastuzumab antibody molecule here, and of course there'll be loads of them binding to many different HER2 proteins. Okay, right. So, what then does human epidermal growth factor receptor 2 do? Well, basically, it induces cells to divide. It is a growth factor receptor. Okay, when it gets activated, it activates growth in the cells. Okay, it activates the cells to get bigger and then begin the division process, because in order to divide, you have to first get bigger and then you divide. Okay, so, basically, in certain forms of breast cancer, HER2 plays an extremely important role in driving overproliferation of the cancer cells. So in our simple model of cancer, bringing back the picture here, you can imagine that this mutation that caused the overdivision was a gain-of-function mutation in HER2. Okay, and often it's an amplification of HER2, so you end up with more than 2 genes for HER2 in this cell. So what will happen is it will somehow end up getting a 3rd gene for HER2 in some way, and then it will produce far too much HER2, and then the epidermal growth factor that's in the extracellular fluid will overstimulate the cells and they'll start dividing too much. Okay, so that's often what happens in certain forms of breast cancer. Okay, HER2 is very important in driving the overproliferation of the cancer cells, or of these cells that are going on to become cancerous. Okay, and these forms of breast cancer, where HER2 is extremely important in the proliferation, are known as HER2 positive forms of breast cancer. Okay, now, in these HER2 positive forms of breast cancer, Trastuzumab or Herceptin is an extremely effective treatment, because what you can do is give this drug, it will then bind to the HER2 receptors on the surface of the breast cancer cells, and then effectively it acts as an antagonist. Okay, it stops the epidermal growth factor molecules from being able to bind to the HER2 receptors and stimulating them. Okay, so effectively it's just acting as a very, very fancy antagonist, and it blocks the HER2 growth signal going to the cancer cells, and that stops them over proliferating then. Okay, so that is an example of where you look at the tumour genome, okay, to see whether the HER2 receptor is responsible for the cancerous phenotype, okay, and it won't be responsible for the cancerous phenotype in many forms of breast cancer, but if your form of breast cancer does contain some overactivity in HER2, then this drug will be effective in stopping that, and therefore stopping the overproliferation that's being caused by that. Okay, so this is an example of where we can look specifically at your form of cancer, your tumour, and decide whether this drug is going to be effective by looking at the tumour cell's genome. Okay, so it's a good, very good example of personalised cancer medicine. Okay, it's also an example of targeted cancer therapy because it is specifically targeting one of the molecular features of the cancer cells. Okay, right, so we'll call it there for this video. In the next video, what we will do is we'll look at some very similar examples to this. So we'll look at Cetuximab and Panitumumab. Okay, and then we'll go on to the small molecular inhibitors of BCR-ABL, which is another classic example of personalised cancer medicine.